So I'm here to talk about the open access research program or model at SGC. Some of you may not know what SGC is about, so I'm going to start with an introduction. We have a lab based in Oxford. Some of you have already mentioned the Oxford SGC lab this morning. We have one based in Toronto as well. We're funded by a number of Canadian government agencies, the Wellcome Trust, and an increasingly long list of pharmaceutical companies as well. We do structural biology, chemical biology. Some of that work goes towards target validation and discovery. We are very heavily engaged in protein targets that are involved in epigenetic mechanisms. Protein kinases are definitely a strength for us, membrane proteins and parasitology. Over a period of 10 years, we have put over 1,500 protein structures in the PDB. I believe roughly over 1,200 are unique first ever protein structures. My colleagues have produced 18 epigenetic chemical probes. Now that sounds like a very small number compared to the number of structures. However, the criteria for what we define as a chemical probe are very stringent. And there are pharma partners, some of which are represented here, I think will tell you that these are high quality chemical probes which are completely made available on demand. There's a web page on our website that you can go to, and there's a contact information, and you can just simply ask us for the entire set of our chemical probes. In fact, our chemical probes program have been so successful that there are actually chemical companies out there marketing the SGC probe set. But I recommend that you come to our website every so often. You'll find that maybe once every 2 months there'll be a new probe molecule added to it, and you will find all the information about the probe molecules on the website as well. A few years ago, our director, Alan Edwards, published a paper in, I think it's Nature Structural and Molecular Biology, and the title of the paper was Primitive Principles Meet Structural Biology. He was basically describing the fact that one of our guiding principles is we put the structure in the PDB as soon as we solve the structure. Now, obviously, there are validations and various other steps involved, but one thing we don't do, which is a common practice, is hold the structure from the public domain or withhold it from the public domain until we publish a paper. Now that puts us, the scientists, at a disadvantage, but it's something that we believe can be very helpful to others. Not only do we put the structure in the public domain, we disseminate the methods and materials on our website. We fall behind because once in a while because we produce structures at a very fast rate, but for most of our structures, if not all of them, you can find exactly how to reproduce it on our website. If you find anyone that's missing, let me know or let any one of my colleagues know. It is our intention not to mislead. Now, as you know, protein crystallization, more than any other, or more than most scientific methods, it's very difficult to reproduce. We moved across the streets in Toronto roughly 5 years ago, and two of the proteins that I was working on that wouldn't even give me the tiniest bit of crystal on one side of the street gave me instant, without any optimization, better than 2 angstrom crystals when we went to the south side of College Street. So that tells you something about the art versus the science of protein crystallization. But yet, when it comes to how to make the protein and how we crystallized it, all the details should be there. We will share anything we can with you. You ask us for DNA. Usually our guiding principle is we'll give you DNA for proteins that we have solved structures of, but to be honest with you, if you need DNA and I have got it, I've never said no to sending it out to anyone. We will give you proteins if you're willing to wait for us to reproduce it, or if we happen to have it in the freezer. We'll give you our the cell lines that we use to express some of our proteins. Over the years, we have actually develops our own customized E. coli cell line that seems to produce more or express more plasmodium proteins than the standard BL21. Some of you may recall over 10 years ago, it was deemed nearly impossible to use E. coli to express proteins. Still very challenging, but I would say roughly 25% of the time, 20-25% of the proteins we've tried, we've succeeded. As I mentioned before, we distribute chemical probes. I'm referring to the human electrical probes for human proteins. We don't have an official chemical probes program for the parasitology program yet. And of course, we publish papers. Now, so that's the theory. That's how we aspire to sharing our work. In practice, so this is what gets interesting. So as you may recall, I mentioned that we have a fairly long list of pharmaceutical companies funding our research, working with us. So why would they work with us if we share everything possible? So there are certain boundaries which protect their interests. Well, first and foremost, of course, is we work in the early stage of pre-competitive research, so to some extent pre-competitive. So that I think protects them. Second of all, each pharma partner submits us target lists, but they don't see each other's target list. And we, as scientists working at SGC, we don't know if a particular target was proposed by GSK, Novartis, or Pfizer. But when a structure is solved, everybody benefits from it. And compound libraries sometimes are provided by the pharmas, are kept confidential. In other words, one company doesn't know what compounds that the other one has sent to us. In some cases, we don't even know what the compounds are, and we basically sign off on the fact that we will not make any effort to find out. Those compounds, however, will not be used in co-crystallization, because if you work with us, any structure we solve goes in the PDB, no exception. That's one key guiding principle. And so there are cases where if you tell me ahead of time, hey, we would like you to screen or assay a certain compound against a protein you've purified, but please don't crystallize it. Use it in crystallization, we will respect that. We want to work with people as much as possible, but our strongest guiding principle is the structure belongs to the public because we are funded heavily by the public. Okay, and however. Sometimes open access for scientists who need publication can be, I wouldn't say a detriment, but can have some drawbacks. And so occasionally I'll start to bottom up. We've been scooped. We put the structure in the PDB. We work really hard in publication, and suddenly we find out, and of course Murphy's Law says just as we think we're ready to submit, somebody publishes a paper. And one of the frustrating things about that is someone can take your structure in a PDB, refer to it, draw very nice figures to it, but not mention anything about the statistics, and the reviewers have never critiqued the structure. You put your own structure in your paper, you're obligated to cite all the statistics, and there's usually at least one reviewer who finds something wrong with how you've done the modeling, and you spend 6 months arguing, refining the structure. So in other words, sometimes it's easier for somebody else to publish our structure than it is for us. But that's part of the price we pay for doing our research, and it's a very small price because, as I mentioned, this happens very occasionally. We do run into other issues when we try to move forward with collaborations. Someone mentioned before universities, the patent office can be a bit of a hindrance. Well, the same folks also work on MTAs as well, and they're very picky. And I say that acknowledging that at University of Toronto, so sometimes the delays at my end, University of Toronto can be picky as well. And I would tell you with all honesty that pharmaceutical companies have never been a problem when it comes to MTA because they're consistent, they work efficiently, and so really that may be contrary to what people perceive. We understand and appreciate that whether our partners, our collaborators are from industry or from academia, there is interest and sometimes an obligation and a need to file for patents. So that does create a bit of an issue. But I don't think there's been one instance where it is important to work together that has been showstopper. Sometimes we get into long-lasting discussions, but we work around that. So now I want to just zoom in a little bit more on to what my team does, which is what I refer to as structural and chemical parasitology. And so this is one of the few times when I talk about my program without really getting into structure. So we basically do the same thing as my colleagues do who are working on the human proteins. We make the protein, we use either E. coli or insect cells to produce the protein, and we try to crystallize it, we screen the protein against compounds. The idea is to help, to enable collaborators and others who are actually working on anti-parasitic drug discovery or working on functional research. They may be interested in what a protein does. This work is carried out in the University of Toronto. I won't go through the list of diseases and parasites here, because in this audience I don't think you need to do more than just have a quick glance at it. I'm not going to go through all the accomplishments. A lot of it is just basically as some people say, stamp collection. But one thing I want to mention is two things I'm proud of, I should say. One is I've underlined here the methods and materials. I'm repeating myself here. They're published on our website. And I believe it has helped people tremendously. Sometimes the hardest thing is to convince people, yes, it's all there. I'm not lying when I'm telling you I'm using the cell line. Some people don't believe how simple our methods are, but I believe simplicity is the reason why we've succeeded in many cases. And then the second thing that I'm really proud of is the bottom line here. I don't know if you can all see with the microphones here. Basically, our work has generated enough interest that on a weekly basis we're sending out DNA, proteins, and in some cases compounds to people who are interested. As I mentioned before, our initial guideline or guiding principle is to send DNA out when we solve a structure, but the demand has grown such that if I have it, we've cloned it, it's yours for the asking. We actually have an official program or official partner for distribution of DNA, because we have to sign this over to AdGene every time we have a new set. So I've just resorted to, if people ask me, I will arrange for you to have the DNA. If sometimes there's a delay, please keep bugging me, because the demand is sometimes quite frequent, but it's something that we don't want to stop doing. I think very soon we'll find a way to maybe turn this over to MR4 or a similar type of organization to make this easier for us. I'm going to zoom in a little bit more deeply into parasite protein kinases. Now, hopefully, it'll be evidence why it's a good exemplar of what open access research can do. As it was just mentioned in the last presentation, MMV funded or oversaw or funded the screening of, I didn't even know, 6 million plus compounds against Plasmodium. Over 25,000 hits, correct? Yeah, my number, I guess, is incorrect. It's over 25,000 hits. I believe there are now 2 clinical candidates with more coming, and we heard, I guess, the introduction of a new preclinical candidate this morning from Dundee. So this has been a very successful program. I think Paul also mentioned, well, why didn't people take advantage of all the published information immediately? Well, the number is quite, I think, bewildering. If you look at the fact that the Plasmodium genome has over 5,000 genes, we're talking about now 25,000 compounds. The number of possible permutations you can work with is just beyond anyone's capability. So we all, based on our background and interests, find ways to narrow down that search space. MMV has used cheminformatics and I guess chemistry expertise to narrow it down to 400 compounds, and that's a great program. And we will soon provide feedback and hopefully publish what we have done with those 400 compounds. We've also, another way is based on what your interest, what enzymes or proteins you're interested in. You can narrow it down to your specific pathway of interest. For example, that's not guaranteed to lead you to an essential druggable pathway. pathway, but that's another way to narrow things down. So we try to look at it from a rational perspective. Roughly around the same time, or maybe shortly after the papers on phenotypic screening started coming out, there was a paper in Nature on the Plasmodium kinome, listing basically listing the close to 40% or so of the Plasmodium kinome being essential, and 2 others essential in transmission. And in addition, there was a breakdown in one of the papers, particularly the paper with the about the screening done by GSK, where of all the hits that they published, roughly 1/3 of them are putative kinase inhibitors. And we at SGC we have a very, very strong human protein kinase program. And at that time, we were fairly interested in parasite protein kinases already. We already started working on it when we saw that basically these two publications. We just thought, you know what, we're just going to redouble our efforts here because it makes sense. Because suddenly you're narrowing down that search space from 5,300 times 20,000 or 25,000 to roughly 40 by 5,000. Still very large search space, but substantially reduced. So how are we tackling the parasite kinases? Well, first of all, now that we narrowed down the search space, we expanded it again because we're not just interested in malaria kinase or Plasmodium kinases. Without question, the true neglected diseases do not benefit from nearly the amount of, or even I would say proportional amount of funding. So one way to try and I think sort of tilt the table back a little bit, the other way is to take research done in Plasmodium and translate it to other diseases. So every time we think of working on Plasmodium kinases, we try to work on orthologous kinases in Cryptosporidium and Toxoplasma, and we're also interested in Trypanosoma and Leishmanian kinases as well. So essentially, we try to go after expression purification of these kinases. When we screen the malaria box, and other kinase inhibitors we can get our hands on, but I want to specifically mention the kinase inhibitors from GSK. Some of you may have heard of the PKIS, the published protein kinase inhibitor sets that's been released by GSK, I think, starting about 2 years ago. We also got Novartis and Roche to share with us some representative compounds from their kinase libraries. So suddenly now, we went to some rational steps of narrowing down search space. But once we did that, we tried to expand it again so that we can take advantage of what I would call medium throughput screening capacity that we have. I want to emphasize the PKIS, GSK's PKIS compound set here. You can find it in Campbell. You can find it in PubChem. You can also contact, in particular, Bill Zerker and Dave Drury at GSK in North Carolina, and they will send you compounds that PKIS set once you explain to them what research you're interested in. So in other words, sort of same type of approach that MMV is taking with the malaria box and I guess soon the pathogen box. And what GSK's interest is that they want to generate as much data on these compounds as possible. So whether you're working on human protein kinases, are you working on parasite kinases, they're interested in supporting you in that research and also getting the data back so that they can really broaden their understanding of kinase selectivity, which has turned out to be a much harder problem, I think, than anyone anticipated. Even though we're working on multiple kinomes, I always like to come back to this Plasmodium kinome tree, kinase tree, as sort of as a reference. And also, I think in the context of this conference here makes sense because if you know Oliver Bilker at Sanger, he actually gives out this tree which he drew, I think, in PowerPoint by hand because he didn't like the way the software generated the actual tree. He shares it with people on demand, and people sort of tweak the tree to represent what they want to. So I call this the open source kinome tree. And some of you who work on kinases will remember that the human kinome tree also has been similarly well used as well. So these circles here represent the structure, the kinases which have structures. I won't go into that too much. I also want to mention there are 3 Leishmania map case structures as well, 2 of which we solved. Just a little bit of showing off, a little bit of stamp collection stuff. These are some subset of our kinase structures. So quickly to wrap up, so this started with sort of to give you one perspective. MMV and partners dumped a whole lot of data, made available for anyone who's interested and they'd help us drill down by narrowing down that very large chemical space to 400 compounds. Roughly the same time, it may be just as a coincidence, GSK released a PKIS. Novartis and Roche follow up with their set of compounds, kinase inhibitors that they're willing to share. And that's sort of, I think, everything sort of coalesced just right in terms of timing to allow us to I think, really broaden our research on parasite kinases. We found nanomolar hits without doing any chemistry against these targets. We'll be publishing that data very shortly. We like to think that we're giving back by putting data in public domain as soon as we get it, at least the structures. As I mentioned before, by sharing reagents. We also like to think we're giving back by repurposing some of the research that we're doing in Plasmodium into other protozoan parasite kinases as well. I'll wrap up here.